Hello, my name is Joshua Mugabi. I welcome you to DNA analysis. DNA molecules are very long. So a DNA molecule is made up of millions of nucleotides. And nucleotides, we say that they are found in the chromosome. A nucleotide is made up of a phosphate, it's made up of a sugar, and it's made up of a base. That is what makes up a nucleotide. So very many nucleotides, when they come together, they form what we call a DNA molecule. So the DNA molecules are very long, and they may consist of millions of base pairs. They consist millions of base pairs. The base pairs that we are talking about, for example, here we have guanine pairing up with cytosine, and here we have thymine pairing up with adenine. Those are the ones that we call base pairs. So DNA consists of millions of base pairs. In order to study the structure of DNA, the molecules are broken up into smaller fragments by an enzyme called restriction enzyme. So when we want to study the structure of DNA molecules, they are broken down into smaller fragments by the enzyme called restriction enzyme. The restriction enzyme occurs in bacteria, and it protects the bacteria from viruses by cutting up the virus DNA or RNA. So the restriction enzyme that we can use to cut down the DNA molecules so that we can study it is found in the bacteria. And this restriction enzyme is used by the bacteria to protect it from viruses. What the restriction enzyme does, it cuts up the virus DNA. E. coli R1 is extracted from Bacterium Escherichia E. coli, or what we call E. coli. That's where we extract E. coli 1, which is our restriction enzyme. The restriction enzyme do not break up the DNA molecule randomly, but it cuts it at particular sites. So the restriction enzyme is not going to break up the DNA randomly anyhow, but what it does, it will cut it at particular sites. There are particular sites where the restriction enzyme will cut the DNA molecule. For example, restriction enzyme called E. coli 1 recognizes the base sequence CAATTC, that is cytosine, adenine, adenine, thymine, thymine, cytosine, and cuts it between the A's. So this restriction enzyme it is going to look at the molecule, the DNA molecule, and it will look for that area where there is cytosine, adenine, thymine, and cytosine. Then it will cut between, it between the A's. So the restriction enzyme, that one we call ECOL1. For it recognizes those base sequence of CAATTC, and it cuts it in between the A's. Then now it will cut it, and they will become now separate molecules of DNA. Other restriction enzymes, these ones can cut DNA at different places, so produces fragments which are easier to analyze. So you can have other restriction enzyme. For them, they can cut different fragments. For example, others can cut between G here, G and C, and you get that fragment. And then you also get this fragment. It comes also here and cuts between G and G. You get that fragment, and then you also get that fragment. Then now you can analyze those fragments easily. The fragments cut by the restriction enzyme are called restriction fragments. So those restriction fragments are the ones that have been cut by restriction enzymes, and it is the restriction fragments that we are going to analyze. The fragments can be separated using gel electrophoresis. They are separated and can be separated by use of the gel electrophoresis. Now, for example, we have got these fragments and we have cut them using restriction enzyme. When the restriction enzyme cuts them into these restriction fragments, now we can separate them by using gel electrophoresis. The different sized fragments are separated by a process called gel electrophoresis. We are now going to separate the different fragments by that process called gel electrophoresis. Now, the separation takes place in a sheet of a firm jelly-like structure or jelly-like substance, which we call the gel. Now, when you look at our gel electrophoresis equipment, you will see that we have a sheet at the bottom. That sheet at the bottom is what we call the gelatinous sheet. It is called the gelatinous sheet. It is that sheet downwards, the base, or what we are looking at the bottom. Now, we have the smaller holes in the gelatinous sheet. Those smaller holes are the ones that we call the wells. We call them the wells. These are the wells. It is where we are going to put our restriction fragments which we need to separate. We put them in the wells, which are depressions which are found in the gelatinous sheet. Then now, on top of the sheet, gelatinous sheet, we put a solution. We put a solution. The solution is the one which is covering this area. The top area that you're seeing is covered by a solution. And then now we can use a needle to put our DNA extracts into the gel electrophoresis. The sample of DNA extracts are placed in shallow cavities, which we call the wells, cut into one end of the gel. We have already seen that these holes are the ones that we call the wells. It's where we are going to put our DNA extracts, which contains the restriction fragments that we do want to separate. We put them into the well. A voltage is applied opposite ends of the gel. So we are going to apply on our gel, we shall apply the voltage. We apply the voltage supply, and then the other side we put the negative electrode, and this one is connected to this side, a positive electrode. And we remember we have a thin slab of gel, which we call the gelatinous plate down, which has got a DNA, well, where the DNA sample placed in the well cut in the gel, and then the other side the plate is negative and this side the plate is positive. Now we shall put our DNA samples in the well. The DNA fragments, they move from the negative to the positive. So those DNA will move from this side, which is negative, moving towards the positive plate. So the DNA has a negative charge and it moves slowly towards the positive end. Now, since we have placed our DNA extract in the well, cut in the gel, but the DNA extract has got a negative charge, so it will move slowly towards the positive end of the slab. The shorter fragments, these ones travel through the gel faster than the longer fragments. So when you have put the fragments here, the shorter ones are going to move faster than the larger ones. The larger ones may be moving very slowly. Larger ones can be moving very slowly, moving towards the positive electrode. Separation of the fragments may take an hour or more depending on the voltage applied. So as the fragments go on separating, it is going to depend on the voltage applied, and it may take between an hour and more depending on the voltage that has been applied. A sample of shorter DNA fragments travel through the gel faster than a sample with larger fragments. So if here, this is our well where we placed the DNA samples and we want them to be separated. we shall see that those ones that are shorter fragments, DNA fragments, will travel faster than the larger fragments. When they are moving, you will see that the shorter one, they move at a faster rate than those ones which are larger fragments. Now here we have this one. This one is a larger fragment compared to this one, which is a shorter fragment. So this one travels faster than this one because this one is a shorter DNA fragment and this one is a larger DNA fragment. The appearance of the separated fragment on a gel. So this is how we are going to see the appearance of the separated fragments on the gel. At the bottom here, we placed our well. We had our well at the bottom here, and then here, and then here. And then the fragments started going on separating themselves. The lighter one separated themselves by moving faster. Then those larger ones, for them, they moved very slow. So that's how they move on the gelatinous sheet. These bands, they will contain shorter DNA fragments. So the bands that we are seeing this side, on the upper side, they are the bands that contain or have shorter DNA fragments. Then these bands, they contain longer DNA fragments. The bands now we have at the lower sides, these are bands or the DNA fragments that contain larger fragments. So for them, they travel at a slow rate compared to those ones with the shorter DNA fragments. DNA analysis can be used to catch criminals or establishing parentage, finding how closely organisms are related, and many other applications. So we can use the DNA analysis to catch criminals. It can be used to catch criminals, and it can be used to establish the parentage, to show whether the child belongs to a particular parent. It can be used to find out how closely the organisms are related, or it can also be used for many other applications. The pattern of the bands in the gel electrophoresis is known as genetic fingerprint or genetic profile. So those bands, the pattern of the bands, for example, this pattern of the band that we have here, that pattern of the band in the gel electrophoresis is known as genetic fingerprint or genetic profile. That is the pattern of the bands in a gel electrophoresis. We call it genetic fingerprint or genetic profile. So this one is a genetic profile. This one is a genetic profile. Here we have a genetic profile. Here we have a genetic profile. We have a positive here, meaning that the positive electrode is this side. As we said that DNA molecule has a negative charge, so it moves towards the positive electrode. If a genetic fingerprint found in the sample blood or other tissues at the scene of crime matches the genetic fingerprint of a suspect, this can be used as evidence. So here we have different genetic fingerprints or genetic profile, and we have a victim here. This is the victim and this is the genetic profile of the victim. Then we have the sample found at the crime scene. We have the sample found at the crime scene, and this is the profile of the sample found at the crime scene. Then we have suspect 1, suspect 2, suspect 3. We get the genetic profile of suspect 1, we have suspect 2, suspect 3. We want to find out which of these suspects was at the crime scene. Remember, we have got the profile of the victim, and there is another profile that we found there at the crime scene. So there are suspects that we've got, and we have got also their profile. So we are going to use those genetic fingerprints to find out the suspect, and we can use that genetic fingerprint to help us to identify the suspect who was at the crime scene. A DNA sample can be obtained from the suspects using blood. We can use cheek epithelial cells taken from the mouth lining of the suspect, or even the cells clinging to the root of the hair. So we can get the DNA sample from the suspect by using his blood or her blood. We can pick it from the suspect's cheek epithelial cells. It can be got from the mouth lining of the suspect or the cell clinging to the root of a hair. More than 20 fragments from suspect 1, they match those got from the crime scene. So when we come to analyze here, this is the fingerprint that we got from the crime scene, and these are the ones for the suspect. So we have suspect 1, suspect 2, suspect 3. And then now we start to analyze the fingerprint or genetic profile we got at the crime scene, and the genetic profile, the fingerprint profile that we have got from suspect 1. When you look, this band corresponds with this one. This one corresponds with this one. Those are 3. Those are 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. You find that about 20 fragments of the suspect 1, they match those that were taken from the crime scene. So we shall take suspect 1 to be the suspect who was at the crime scene. When you look at suspect 2, you will see that there is no much correspondence. The bands are not corresponding with this one. Then when you look at also suspect 3, you will see that there is no response, or with the fingerprint that we got at the crime scene, there is no any correlation. So our suspect is suspect 1, is the one who might have been at the crime scene. Evidence from the genetic fingerprinting is now telling us that the genetic fingerprinting is a powerful evidence in criminal trials. But many restriction fragments may be crowded into a single band. That is one of the problems with the genetic fingerprinting. You may find that many of the fragments may be crowded into a single band so that you cannot really analyze them very well. There may be a variability in the speed with which the fragments travel through the gel. That one can also be a problem, whereby some fragments are moving very slow while others are moving very fast. There is also a chance of contamination with foreign DNA, like that one from the bacteria. That one can also end up giving us false results if there is contamination from foreign DNA, like for example from the bacteria. The jury may not understand the significance of the genetic fingerprinting and may be dependent on conflicting claims from the expert witnesses. So sometimes a problem may be as a result that the jury does not understand the significance of the genetic fingerprinting. So you will find that they will mainly depend on the conflicting claims from the experts or witnesses. There may be argument about the statistical significance of a match and the DNA profiles. So that one is also another problem, the statistical significance of the match between DNA profile. There may be argument about that statistical significance. Limitation of DNA evidence. Some of the limitation of DNA evidence. 1. Even if there is agreement about the match between the suspect's DNA profile and the forensic samples, it shows only that the suspect was present at the scene of crime and does not prove that he or she committed the crime. So by showing that the DNA profile which was got from the crime scene corresponds with the DNA of the suspect, that one is not any evidence that the suspect committed that crime, even if the DNA is matching his genetic profile. Another one problem is that the DNA evidence should be considered as conclusive proof of guilt only if there is other supporting evidence. If we have other supporting evidence, so this fingerprinting DNA profile, genetic profile, can act as a conclusive proof of guilt only if there is supporting evidence. There has to be other supporting evidence. So it cannot be used alone that this profiling, genetic profile, is a conclusive proof of guilt of suspect 1 that he committed that crime, unless there are other supporting evidence. In case of paternity disputes, the genetic evidence can be conclusive. We can use genetic evidence to be conclusive in case of paternity disputes. The paternity can be decided on the basis of single restriction site. It can be decided on the basis of single restriction site. Now, a child will receive one copy of restriction fragment from the mother and one from the father. So let us first of all understand these fragments here. Here we have the mother, and this is the mother's fragment. She has these two fragments. One has this restriction fragment and another one has this one. Then we have, this is the father, and these are the restriction fragment of the father, parts of DNA strands. So the father has this restriction fragment and this restriction fragment. So what are the possibility of the children of these two? This child might have got this one from the mother and this one from the father. There might be a possibility that a child may get this one from the mother and he gets this one from the father. There is another possibility that a child may get this one from the mother and he gets this one from the father. And then there is another possibility that a child may get this fragment from the mother and this fragment from the father. So those are the four possible fragments that can be obtained by a child from the father and the mother. So it could be any one of these combinations. It could be any one of these combinations. We have this combination here. That one shows that this child is from these parents. It can be this combination here. Even this combination shows that still it is coming from those parents. It can be this combination. This one also shows that it is coming from those children, and this combination is also coming from that one. So the child should have any of those four combinations. The vertical line should not be confused with the chromosome. These vertical lines that we are having here, let us not confuse them with the chromosomes. They each represent the length of a double-stranded DNA in a single chromosome. So this one is just showing the length of the DNA in a single chromosome, but it is just showing the position of restriction fragment in the DNA. Genetic fingerprinting. There is a match between one of the child's restriction fragment and one of the mother's. So let us look at this one. We have here the mother's DNA. We have the child's DNA. And then we have the possible fathers. There is father A here, and there is also father B. We want to find out of these two, father A and father B, who is responsible, who is the father of this child. So we are looking that there is a match between one of the child's restriction fragment with that one of the mother. Now when you look at the child's fragments, these are the child's fragments, and this is the mother's fragments, and these ones are for the possible father A, and these are for the possible father B. But when you look at the child has a restriction fragment which corresponds with one of the mother, which indicates that this child is from this mother. Then there is also a match between the child's other fragment and one of the possible father A. So when you look at still the child, we have already seen that this one, the fragment of the mother, matches with the fragment of the child. Now when you go up, you are going to see that this fragment of the child corresponds with the fragment of father A. Now when you look at it, it means that father A is the father of this child because they have a corresponding fragment. Neither the child's restriction fragment matches the possible father B. Now when you look at possible father B, you will see that his fragments are down this side, and these fragments, they do not correspond with the child's restriction fragment. Therefore father B is not the father of this child. Genetic fingerprinting still, we find out that 90% or more DNA does not carry nucleotide triplets that code for proteins. So during genetic fingerprinting, you find that some of the DNA does not carry nucleotide triplets that are used to code for proteins. So for example, if we have the DNA here and this one here, it may have triplets, but some of them do not code for a protein. The non-coding DNA is called junk DNA, but this only means that its function have not yet been discovered. For example, if you look here, this one, all of this one here, this part of DNA does not code for any protein. It is not used in the formation of any protein. It does not have any coding, so it is called a non-coding DNA, or sometimes called a junk DNA. Some of the non-coding region consists of repeated sequence of nucleotide. The non-coding, you will find that they consist of repeated DNA, they consist of repeated sequence of nucleotides. For example, you see here, there is CATG, then it repeats again, CATG, CATG. It is repeating. So most of the non-coding regions, they consist of repeated sequence of nucleotides. For example, CATG. You have another one, CATG, 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 which are the same as these ones here. Those are repeating non-coding regions. So that is the example. The number of repeated sequence may be much greater than this, perhaps consists of thousands of more base pairs. So the non-coding region may be containing more base pairs in thousands. The number of repeats in any one section of DNA varies from one individual to the next. So the region of the repeats is going to vary from one person to another. So you can see that this is the repeat in this person. And you may find that this person now has got again this repeat. So you can see that the length and the number is varying from one individual to another. Since these sections do not code for a protein and therefore are not genes, there is no observable difference in these individuals. Because these ones are not coding for any protein and they are not genes, so there is not going to be a difference between this individual and this individual, because these non-coding regions, they are not genes and they are not coding for any protein. Particular repeat sequences can be cut out by restriction enzyme. So the restriction enzyme can cut out this particular repeat sequence. So it can come and cut here, and then it can also come and cut here. When this one, it can come and cut here, and then it also comes and cuts there. So the restriction enzyme will cut out those repeat sequences. Let's look at the Human Genome Project. Human Genome Project. An organism's genome is the entire genetic makeup of that organism. So what we call the Human Genome Project, or the human genome, or the organism's genome, it is its entire genetic makeup. The genome includes the following. 1. All the chromosomes, that is the genome. It includes all the chromosome. It includes all the genes on the chromosome. And then thirdly, it includes all the DNA of the chromosome. So what we refer to as the genome of an organism, it includes all its chromosomes, all its genes on the chromosome, and all its DNA of the chromosome. The Human Genome Project set out to 1. To identify the genes in the human genome. So the Human Genome Project sets out to find out the total genes in the human genome. And these are about 25,000 genes, the total number of genes that are found in the humans. They are around 25,000 genes. And it is also used to discover the sequence of base pairs. The human genome is used to discover the sequence of the base pairs. And those base pairs are about 2.8 billions. The sequence of the base pairs, they are about 2.8 billion in the Human Genome Project. 99% of gene containing part of human DNA have already been analyzed by 2003. So the genes, we say that there are around 25,000 genes in the human genome. 99% of the genes have been already analyzed. The Human Genome Project was established in 1990, and was completed in 2003. But analysis of the data still continues. The number of other organisms have had their genome sequenced. Other organisms also their genome have been sequenced. Mapping. Mapping is the identification of genes and their position in a chromosome. So what we call mapping, this is the identification of the genes and their position in the chromosome. For example, here, if you look at our chromosome here, by genetic mapping, this is chromosome 7. It is mapped and found out that at this position here, that is where we are likely to find the gene for cystic fibrosis. And when you look at chromosome 11, it was mapped and it was found out that at this position here, it's where the gene for sickle cell anemia is located. So mapping is identification of genes and their position in the chromosome. Modern biochemical techniques are used to identify the genes and their position in the chromosome. So they are using modern biochemical techniques to identify the genes and their positions in the chromosome. Special staining methods reveal bands in the chromosome. So these bands that we are seeing, the bands that we are seeing in the chromosome, these ones are obtained by using special staining method, which reveals those different bands in the chromosome. These do not necessarily represent genes, but they help to identify the position of the genes. These bands that we are seeing that have been stained, they don't necessarily represent that that is a gene. That's where the gene is located. But they can help in identifying the position where the gene is located. Currently, the position of approximately position of 19,599 genes have been identified. So the position of about 19,599 genes positions have been identified on the chromosome. So those genes, they know where they are located and their position on the chromosome. By special staining techniques, bands appear in giant chromosomes of fruit fly. We have a fruit fly chromosome here, which has been stained, and it can show the position of different bands in the chromosome. Special staining techniques, bands appear in the giant chromosome of the fruit fly. We have seen those giant chromosomes having the bands after staining. The giant chromosomes are from salivary glands of the fruit fly, which we call the Drosophilas, and are thought to be formed when the chromosome fails to separate after repeated mitosis. So this is a single giant chromosome. It is very long because of failure of the chromosome to separate after repeated mitosis. The bands do not necessarily represent genes, but if a mutant flies, in mutant flies, some of the bands are missing, there is a corresponding defect in the fly. So these bands that we are seeing, they may not necessarily represent the genes, but in the mutant flies, these bands can help us to look at the missing band, and then it can help us to identify the defect which is got by that mutant fly. For example, if this band is missing from this region, the fly has no color in its eye. It is normally red. Now, if this band is lost, it leads to the change in the texture of the eye surface of the mutant fly. If this band is missing, there is an irregularity in the wing. If this one or more of the bands in this region, they control the normal development of the bristles. Sequencing aims to find out the sequence of the nucleotide in a stretch of DNA. Sequencing is going to help in finding out the sequence of nucleotides, or where the nucleotides are in a stretch of DNA. The process can be automated to give results very quickly. Sequencing can be automated. This will give results very quickly. Analysis of a small piece of DNA might give results something like this. Further analysis is needed to decide which sequence codes for proteins and represents the genes. So we can analyze the sequence that has been observed, and we see which sequence can code for a protein and which one represents the genes. Application of results from human genome projects. How are the applications applied? It is hoped that the knowledge from human genome will enable the identification of defects, of defective genes, and the chance of early treatment. So by carrying out human genome project, it can help us to identify the defective genes and the chances of treating those genes early enough. The human genome enables the identification of genes which could make a person susceptible to certain diseases. So the human genome, knowing the human genome, will help to identify some of the genes that can cause a person to be susceptible to a particular disease, and so it leads to preventative measures. Human genome also enabled in prediction of the proteins that genes produce, giving the opportunity to enhance or inhibit these proteins by specially designing drugs, So human genome can help us to predict the proteins that are produced by particular genes, and it gives us an opportunity to enhance or to inhibit this protein by especially designed drugs. Among the possible drawbacks of human genome project are the possibilities that insurance companies may refuse to cover people at risk of developing genetic disability or diseases. One of the problems of the Human Genome Project is that when the genes have been found out and people have got those genes, insurance companies may refuse to cover those people who are at risk of developing genetic disabilities or diseases of those people who have got those particular diseases that have been identified. And then another drawback still of a human genetic project are the possibility that the prediction of a disease or disability could blight a person's life. Hello, thank you very much for participating in DNA analysis. Please do like, share, and subscribe.